Let's take a look at the reason for the banding pattern in skeletal muscle. So to look at this, first remember that a skeletal muscle cell is filled with myofibrils and that the sarcomeres of these myofibrils align within the cell. The banding pattern that we can see from the surface of the cell is due to the pattern or the arrangement of thick and thin filaments in the myofibrils. And so let's take a look at this. Wherever the thick filaments are, and I'm just going to draw a dotted line at the boundary of the thick filaments. You can see the thick filaments highlighted in red are in between my lines. Basically, wherever the thick filaments are, this whole area will appear dark when we look at it under the microscope. So taking a look at the neighboring sarcomere, I can see this is where the thick filament starts, and then of course the end of the thick filament is cut off there. But where I see where thick filaments exist, using light microscopy, I'm not going to be able to see thick filaments. They're way too small. I just see that it looks dark. Okay, again, let's look at the other neighboring sarcomere. So there's the boundary of the thick filament, and the other boundary is cut off, and so where there are thick filaments, it appears dark. And so that dark band that's created is known as the A band. Okay, so the A band is the dark band. Now let's take a look at what doesn't appear dark. What we have is, here's the boundary of one sarcomere to the next. Okay, here's the boundary of one sarcomere to the next, right? So this is one sarcomere. So I can see that the lateral parts of neighboring sarcomeres do not contain thick filaments. And in that region, it appears light. That's what's known as the I band. The I band is the light band, and so what we can see from the surface of the cell is this alternating A band and I band. A light band, a dark band, light band, I band, and so on. All right, let's take a closer look at the A and the I bands because there are some other features that we can see. So we understand that where there are thick filaments, that in between these lines, this is just going to appear dark, and that's what we call the A band. I'm not going to fill this in yet because I want you to see how the thin filaments, remember, come in from the side of the sarcomere and they partially overlap the thick. They don't make it right to the middle of the sarcomere, but here you can see the thin filaments shaded in blue. So let's just take that into consideration for a minute. So when thinking about the A band, what we can sometimes see, even with a light microscope using oil immersion, is sometimes you can see like where the thin filaments are overlapping. So in other words, in these areas where the thin filaments are overlapping the thick, that this area, will appear, even darker than the center of the A band. Okay, so again, over here where we have overlap of thin filaments and thick filaments, that's going to appear the darkest. Now, where the thick filaments are located, this is still all called the A band, just like we said. All I'm trying to point out now is that sometimes you can notice this region right in the middle of the A band where it's a little lighter. That's because there is no overlap of thin filaments and thick filaments. It's just thick filaments, and so it appears slightly lighter. And that's called the H zone. The H zone is a lighter region right in the middle of the A band. And then in addition to that, using an electron microscope, you can actually see the protein that's anchoring the thick filaments in place. And that line that's right in the middle of the H zone is called the M line. And the M line is just the line of protein that's anchoring the thick filaments in place. So in summary, what we can see in the A band is what we can sometimes see is a lighter region called the H zone that's right in the center of the A band. And then right in the center of the A band and the H zone is the M line, which is the protein that anchors the thick filaments in place. Taking a look at the thin filament, and the I band. We know that where the thick filaments are not located, that's going to appear lighter. Okay, so in other words, we just looked at the fact that this is the region that we would call the I band. The I band is, half of one or a little bit of one sarcomere and a little bit of the neighboring sarcomere. And right in the middle of the I band is the actual boundary from one sarcomere to the next. And so this zigzag line is the boundary from one sarcomere to the next, but it's also the protein that's anchoring our thin filaments in place. We can sometimes see that using a light microscope. That's called the Z-line, and the Z-line is the border from one sarcomere to the next. So taking a look at another light band, I band, see if this makes sense. So the I band would be where the thick filaments are not. So in other words, this region is what we're calling the I band, and right in the middle of the I band, what we notice is that there's the boundary between one sarcomere to the next, right in the middle of the I band, and that is the Z-line. Let's take one last look at this banding pattern. Across the top is an electron micrograph, so an actual photograph of skeletal muscle, and it's aligned with a diagram of the sarcomere. And so what we see is that where the thick filaments are located, in between my yellow lines, we would call this entire distance the A band. So all of this is the A band. And then we can also see, and you can line this up with the diagram, that where the thin filaments end, that it's a little bit lighter in that region. That's what's known as the H zone. And we can also see a stripe going right down the middle of the H zone, corresponding to the protein that anchors the thick filament in place. That's what's known as the M line. We can see the lighter I band on either side of the A band. And we can see the Z line right in the middle of the I band, which is the border from one sarcomere to the next.